Hi everyone, thanks for coming. My name is Alex Valentine. I completed my Keystone project as a part of the senior design project in the engineering school, and the title of it is Cell Sheet Interaction on Three-Dimensional Bioprinted Tubular Constructs. So there's a pretty significant clinical need in the healthcare industry today. Congenital heart defects affect 1% of newborns, and it manifests itself as abnormal development of heart tissue or blood vessels around the heart, and it often requires, the severe defects often require reconstructive surgery, which will involve implantation of a vascular graft, which is like a synthetic blood vessel to fix the malformation in the blood vessel. So for instance, there could be like a narrowing of a blood vessel. So this is like a narrowing of the aorta in the heart. So it's important to fix these things, and surgical treatment will involve implanting grafts, but unfortunately right now they can lead to complications such as thrombosis and aneurysms, but more importantly they lack in vivo growth, so they're synthetic materials that will be implanted into the infants, but the materials won't actually grow as the infants grow, so their hearts will physically grow, and they'll grow out of the grafts, which is an issue. So tissue engineering has been able to fix this, or is on its way to fix it with something called tissue-engineered vascular grafts. And what this is essentially a lab-grown blood vessel. It will have the growth potential that was lacking in the synthetic blood vessels, and it will actually be able to grow with the infants from when they're born with the defect after surgery happens to when they're children and adults. So what's important is that they can mimic the native tissue, like blood vessels exist in physiology based on how they grow them in the lab, and people have actually already made them and implanted them. So this is a lab in North Carolina that actually made a synthetic blood vessel out of living cells and living tissue and was able to implant it into a baboon and bypass the carotid artery and have it actually flow, which is incredible. But what's important for these grafts is that they maintain structural reinforcement. So my project focuses around adding reinforcement and kind of helping the development of these grafts with two technologies, cell sheet engineering and bioprinting. So cell sheet engineering, it's a really cool technology, and it basically involves growing cells into one cohesive unit where they will act together kind of like a sheet of paper, and you can detach them and stack them and wrap them and things like that and use them for different tissue engineering applications. So you can see these are cell sheets made out of heart cells, so they're beating just like hearts do, and they're able to be physically manipulated and stacked on top of one another. So this is very useful to theoretically be able to wrap around a vascular graft to give it more structural integrity. And also another cool thing in tissue engineering is three-dimensional bioprinting. So this is useful to like make or synthesize a three-dimensional construct out of biological tissue. So I'm sure you guys have heard of 3D printing and things like that. It'll print like, little constructs out of like plastic, but this will print actual biological tissue. So this is a bioprinter that we actually use, and our group actually made this construct. So you can see it's actually printing a tube that would mimic a blood vessel out of biological material that has cells encapsulated in this material. So this is essentially like making a blood vessel and building a blood vessel from scratch, which is a really cool idea. So what we want to do is essentially fabricate and test this bioprinted construct for in vitro culturing of cell sheets. So we want to see, is it useful to use these cell sheets in conjunction with a 3D tube structure or a 3D vascular graft and use them to structurally enhance these vascular grafts. So to break it down, what we need to do is first make the bio-ink that the bioprinter would actually make. Next, we need to actually bioprint the structure, which required a lot of different modifications to the printer. We needed to grow and harvest these cell sheets ourselves in the lab, and finally actually conduct the tests of seeing how these cell sheets could interact and potentially wrap around these bioprinted structures and hopefully create a complex tissue. Something to keep in mind for the bio-ink is that it has to be polymerizable, which essentially means that it needs to be able to take on different material properties. So what polymers do essentially is they can exist in kind of fluid states when they're pre-polymerized, when they're kind of fluid like honey, and then you can change something about them and then make them hard and solid like rubber. So essentially in this case we have a honey-like substance that can undergo a change based on exposure to UV light, and that will make it hard and rubbery. So the material that we chose based on testing in literature is called methacrylated gelatin, or GELMA, so that's going to be the bio-ink that I talk about for the rest of the presentation. So when I say GELMA, that's what that means. grains. And the next more important thing when it comes to innovative techniques that we're employing is the use of a substrate that will actually allow the cell sheets to detach. So when you grow the cell sheets, cells like attaching to whatever they grow on. And it's really hard to lift them off something without killing them. So what's cool about this material is it allows for cell sheets to actually detach after they're grown into one whole unit. The cell sheet is grown. So the material that we grow them on is called NIPAM, and it's a material that actually has properties that will change based on temperature. So if you look at this temperature meter through this animation, as the cell sheet lifts off, or a change in temperature allows the cell sheets to lift off. So when you decrease the temperature to like 20 degrees, it's not long enough to kill the cells, but it's long enough to change the material properties of the surface and allow for the cell sheet to actually be lifted off. So without this special material, the cell sheets would just stick on it, and if we tried to take it off, it would just rip into pieces. So this is a pretty useful part of our project for cell sheet engineering. For our methodology, we needed to test multiple different bioinks when it comes to their mechanical properties and their viability with cells to see if they're actually biocompatible. We needed to modify the bioprinter to work with our bioink and test it to make sure it could actually print multiple materials and print 3D structures. We needed to chemically make that NIPAM substrate, which was actually pretty hard, but we needed to make sure that we were getting an even coating, that the cells wouldn't be growing more on one part than another, and they're growing into one solid unit that would act as like a cell sheet. After we knew that they would actually grow on it, we needed to grow them and harvest them and make sure that they would come off properly. And finally, we needed to test the procedure of transferring these cell sheets using the NIPAM onto a 3D printed substrate. So that's like the whole idea of it. So, to the good stuff. Results. We were able to show that we can print multiple materials at the same time. So this is JELMA printed, just of different colors using food dye. So we can show that we print a red and a blue JELMA at the same time in a specific pattern. So this is just concentric circles. There's 2 here and then 4 concentric circles here. So this just shows that you can print like a very small but complex structure out of multiple different materials. And we're also able to show that we can do this with cells. So here, this is the same exact thing that I showed you before. It just has 2 different materials that you can't actually see because they're not colored, but they actually have 2 different cell types in them, and we can show that we're able to pattern cells using this same concentric pattern in this way, and we can show it with this fluorescent image. So in the same way that blood vessels are organized with endothelial cells on the inside of the tube and smooth muscle cells with the outside of the tube, this is represented the same way, where the yellow are the endothelial cells, so that would essentially be the inside of the tube, and the blue are the smooth muscle cells, which would be the outside of the tube. So this is a pretty significant finding, showing that we can actually pattern cells on our own based on whatever program that we actually give the computer, because most people just grow cells and they just grow however they want, but this is extremely useful. So this is the tube blood vessel construct that we were able to grow, or that we were able to fabricate with the bioprinter. So from that video, this is the result from it. So it's about 8 millimeters tall, and the wall thickness is like 0.5 millimeter. But what's important is that you can actually put it in PBS, which is like a biologically relevant buffer, and let it sit, and it will actually maintain its structure over 3 weeks. So you can see that from the side, it still maintains its height, and it still maintains its lumen. So you can still actually see all the way through it. So that means that it still maintains like the blood vessel structure. But unfortunately, we found that it was too soft to physically manipulate. So when we pick it up with forceps, it will actually collapse under its own weight. So we still need to do some testing and change the mechanical properties of the bioink, but we're still able to show that we can make a blood vessel structure that will float around, which is pretty cool, I think. When we were testing our bioinks, we need to make sure that they were viable with the different cells. So these are so the JELMA we show where the green cells are alive and the red cells are dead. So we show the JELMA as compared to a positive control, which is AlgCol. It doesn't matter what that means, but it's basically a different kind of bioink. And the left column shows smooth muscle cells, and the right column shows endothelial cells. So you can see usually in this diagram a significant amount of live cells. Unfortunately, this JELMA condition, we found that all the cells died because the pH was too low. So after correcting that, this condition actually was This image was taken after this one, after we were able to neutralize it, so we can show that we can indeed use the GELMA as a biologically relevant bioink, and the cells will live in it, which is useful. So we can kind of assume that the endothelial cells will live and proliferate similarly to the smooth muscle cells, which is great. This is kind of complicated. It doesn't really require me to for me to explain all of the details of it, but essentially what it means is we're able to make that NIPAM, that special surface, and grow cell sheets. So under different conditions, sometimes the cells live and sometimes they die. So we use this condition, obviously. For transferring, we're able to show that the cell sheets will actually stay intact throughout the process. So from pre-transfer to transfer to post-transfer, we can show that they do still stay intact on the GELMA and on the positive control. Unfortunately, most of them died, which was because of the actual physical handling of them during the process, but it still shows that we could actually transfer them onto a flat substrate made of the bioink. Finally, the flagship experiment was actually being able to wrap the cell sheets around a tube structure, which we were able to successfully do, which is great. So you can actually see, maybe this is better, you can actually see the cell sheet here with a zoomed in picture, and if you follow the white outline, that represents the cell sheet being wrapped around the physical tube, and you can also see that there are cells viable. So this shows that throughout this process, cells will actually stay viable in the cell sheet when we're wrapping them around this tube construct, which no one has actually done before. So what does this actually show? We've proved that GELMA is extremely valuable as in bioink for bioprinting and for tissue engineering. We've shown that we can bioprint complex tissue constructs, and that's a feasible process to do in 3D and with multiple materials. We've shown that NIPAM will make a great substrate to harvest and grow cell sheets on. And finally, we show that that experimental protocol for wrapping the cell sheets around a bioink structure is actually viable. So with a little bit more work, we can actually make that a process that would be successful with a lot higher viability. So moving forward, we'd like to hopefully optimize the culture conditions in vitro. So we'd be able to wrap the cell sheets and keep them wrapped around that structure for a bit longer and keeping them alive. So maybe we could employ like a rotating culture platform, because right now they kind of just sit and float, which obviously isn't biologically relevant. That's not how the cells would actually live in vivo. We would need to optimize the different bioprinting conditions. So the multicellular pattern that I showed you before, it was cool to show you that they were all patterned. Unfortunately, all the cells died because it took so long to load them and print them in the bioprinter. So it would take some optimizing for the actual bioprinting process to make those cells still viable after that entire thing. And importantly, we've been able to show that bioprinting for tissue engineering has the potential to become a very significant technology because it is so versatile. You can program a printer to print whatever you want. So a tube is just a tip of the iceberg. You can print theoretically whatever you want, which is amazing. I think that's kind of why I got into biomedical engineering, because it's so cool. And actually, 2 labs at Harvard have shown that bioprinting this exact material will be able to create vasculature. So that just further verifies that the concept and the idea behind our project was actually viable and is actually like a good idea, because people are already doing it. They're like beating us to the punch, but it's still good to show that we're going down the right road. And overall, it shows that our studies have a pretty significant potential to inform the future development of cell-based tissue engineering vascular grafts, which will be used to repair vascular malformations in children with CHD. So with that, oh, maybe it's not working. Sorry. There we go. Okay, so last thing. This is just a cool thing that me and my group members did while we were finishing our bioprinting. So this just shows the versatility of the printer and how you can program it to print whatever you want. So this, like my title slide, actually shows that we were able to print, letters like the UKHC out of biologically active material. So they're actually cells in that living, which is just like a cool, fun thing that we did. So yeah. And yeah, obviously, I'd like to thank everyone. Most importantly, my team members. Mike's actually here, and Kevin and Will, who were the rest of my senior design group who made this all possible. And obviously, the Kilachon Arms College and everybody. So yeah, thanks. Questions? I don't know, how am I for time?
I have about 1 minute.
Okay.
What do you think that
I like it will be difficult to. I can understand you can transfer the cells there, but how is it technology there that they will grow and they attach to the target you are going to do there? Because I saw in your cells died.
Right.
This is the most challenging part of this.
That is the most challenging part. And like I said, so the labs who are actually working on this have shown, I didn't put it in my presentation, but they were actually able to print cells that would live in culture, they would survive the process, and they would actually like perfuse blood vessels. So they're a lot farther down the road from us, but it proves that it is viable. And after optimization, it's definitely possible. Yeah.
This is just like a clarification question, but so JELMA was basically like a scaffold for your cell layers to run
Right,
on.
yeah.
And then the thing that you were, like the essential property of JELMA was that it enabled those epithelial cells that you wrap around it to keep living. Okay, so then with past scaffolds that people use, do they not, like past grafts that they're using don't actually use real cells that wrap around it?
Yeah, so the question.
Real cells around those grafts that are being used now.
Right. So yeah, so the question was basically like for people who are making the grafts now, are they growing them on scaffolds that have cells in them or not, right? Okay, so as far as I know, no, and that's what's so cool about ours is that we can use a bioprinter to actually make a structure that has cells in it, so that would make the attachment of the cell sheets like that more biologically relevant.
It'd be cool if you could just get the JELMA to like degrade also, like.
Well, yeah, so that's the cool thing. So she said it'd be cool if the JELMA could degrade. So JELMA is actually just made out of collagen that's been degraded already. So the cells, that's why the cells like it so much, is because it's already like a biologically relevant molecule. But it has sites in it to degrade. So the cells could degrade it over time. Yeah. Sorry, I'm like way over time, but yeah, thanks.